So tēnā koutou katoa. Thanks so much for the warm welcome, Sarah Jane, and thank you for joining us this evening. So as Sarah Jane mentioned, I'm based now at the University of Otago in Dunedin, and I have a specialist research interest in movement neuroscience. In particular, I'm really interested in and have been working for a while now on understanding the way in which emotions, especially negative emotions or emotional stress, influences our motor system. So how emotions might be altering the way in which we generate movement, how it might be altering our ability to control or perform movements under stress. So collectively, I'm interested in our motor behaviours. So this evening, I'm going to share with you some findings from a selection of studies where I've been investigating how stress alters the functioning of the motor system in Parkinson's disease. Before I do, I just wanted to thank the Neurological Foundation up front, not only for the invitation to talk this evening, but for funding the research that I've been conducting in this area since 2015. I wanted to thank the team of researchers at the New Zealand Brain Research Institute, where I was previously based in Christchurch, for the contributions to the work, and some of the specific colleagues I've acknowledged on the last slide. And last but not least, thank you to the incredible pool of volunteers and their families who continue to donate their time so generously to make this research possible. So for the last 10 years, I've been really curious about this interwoven nature of emotion and action. And throughout the talk, I may use the term emotion interchangeably with affect. For the purposes of tonight, these are interchangeable. And I may also use the words movement or motor control or motor behaviour synonymously with action. So I'm curious in understanding which parts of the brain that control our emotions and that control our movement might be interacting together or communicating with each other, and whether or how this might be different in people with movement disorders. So this idea that emotions are intimately related to action has been expressed by emotion theorists like the late Nicol Flyder. And in his view and others, all of our emotions can be expressed as motor behaviour. And if we think about it in the reverse, that all of our actions have some form of an emotive component. And what's quite interesting is that the bridging of these two fields, so affective or emotion neuroscience with movement neuroscience, is very much in its infancy. It's only been over the last 2 decades or so that neuroscientists from either the emotion domain or from the motor control domain have started to unite these two fields to examine how emotions might influence our movement at both the brain level, so our central nervous system, as well as at the peripheral nervous system level. So for example, changes in our muscles. But this idea isn't new, this idea of this close relationship between emotion and action. It's a concept that dates back to the 1800s and is in itself implied in the Latin word for emotion, which you'll see on the slide here, includes the verb to move. There's been an abundance of anecdotal evidence, of course. We only need to think about giving a public talk and our hands trembling. But there's very little in the way of scientific research from a neuroscience perspective. So for example, I'm interested in why when we're joyful or we're happy, do we jump up and down, we embrace people? Why do we lash out and we hit with anger? And particularly how strong or what we call high arousing emotional contexts might alter our motor control. So this unique situation of Olympic competition, for example, enables some athletes to boost their motor performance and they break records. Other athletes can choke. Understanding how surgeons can maintain precise control over their fingers when they have these really fine motor control movements, despite this potentially really stressful environment. Why, when we're sad or depressed, do we walk with a stooped posture, or our gait speed, our walking speed is slowed? Understanding how anxiety can hasten our movements, we end up trembling. Or fear can leave us frozen. Our muscles can become paralytic. And there's been renewed interest in how stress may be exacerbating motor symptoms of Parkinson's disease and how it may even have the potential to accelerate neurodegenerative processes associated with neurological disease. So my research took me to the University of Geneva, where I spent 3 years learning neuroimaging. And I conducted a number of studies using functional magnetic resonance imaging. So the MRI machine is, you may have had an MRI before, it doesn't necessarily have to be on your brain, it could be on another body part. It's exactly the same equipment, the same technology. It allows us to look at the structure of the brain, much like an X-ray. But the way that I use this piece of machinery is to look at changes in the functioning of the brain. So we can get participants inside the scanner, we ask them to do a movement task, and we look at changes in brain function or increases or decreases in activity of the brain during that task. I then came back to New Zealand and spent 5 years at the New Zealand Brain Research Institute in Christchurch, and here I was combining neuroimaging with a range of other methodologies. And all of these allowed me to look at movement in the upper limbs. So I'm mostly interested in motor control of the hands and the digits. So we look at skilled actions like grip force, and we also look at changes in tremor. So for grip force, here we have this little device, and we ask participants to squeeze with their index finger and their thumb using precision grip. And this allows us to look at or measure the amount of force that people can produce. And we can look at various aspects of force production. We can ask people to generate a maximal squeeze. How hard can they possibly produce this action? Or we can ask or examine how people can control low levels of forces. And we look at this type of action because these are functional movements inherent in everyday activities. We use grip force to carry our shopping bags, to grip our cup of tea, to write with a pen. And grip force is what we call skilled action. So it requires fine motor control and the precise coordination and timing of numerous muscles in the hand and in the forearm. We also know that grip force is an action that's affected in a range of movement disorders like Parkinson's disease and like stroke. In almost all of my studies, I use electromyography. This is just a fancy way of saying that we look at muscle activity. So this is non-invasive. We have little devices called electrodes that sit over the surface of the skin, and they pick up the electrical activity in the muscle when the muscle is contracting, when it's active. So here we can look at the amount of muscle activity, and we can look at the timing of that muscle activation. So when a muscle is active and when it relaxes again or it goes to rest. And the other method I've been using is accelerometry. So as it's implied, this is a little accelerometer that we tape over the participant's finger, and it allows us to measure those little accelerations that we see in the fingers during tremor. And this doesn't have to be Parkinson-only tremor, this could be essential tremor and other disorders. And accelerometry in a clinical setting is very much the gold standard method to assess tremor severity, for example. So data from all of these measurements tools I'll present today. So I'm going to present a snapshot of results from a couple of studies that all come under this umbrella question of how do negative emotions affect our motor control? And the first study is looking at grip force, and it was conducted in Geneva in healthy young volunteers. So this is like our student university population. So I'll just go through the task or the study here. So we asked participants to squeeze on this grip force device with just a low level of force. So it's 10% of their maximum. So they don't get too fatigued, and they do this for multiple trials. So when we measure from this force device, we get a trace that looks like this. So here they're just still relaxed. Then they get the instruction or the go signal to start squeezing. And we ask them to try and maintain the same level of force as best they can. So they're trying to be really steady, keeping on that target level for about 12 seconds at a time. And then they relax and wait for the next trial. Participants are doing this while they're watching a computer screen. And on the computer screen, we're presenting to the participants emotional images. So I'll just take a little bit of a sidestep and talk about the way in which we can examine emotional state in a lab or a research environment, and how I've been using these emotional images to induce an acute stress response. So there's lots of different methods you can use. I use these images that I take from an emotion database. There's about 2,000 or so images that you can choose from, and depending on how you choose them and categorize them, you can look at various things. So I look at two different categories or conditions. So we have a bunch of photos that are considered pleasant. So they're pleasant images to look at, pictures of babies and puppies, sunsets, and so forth. So these images comprise our non-stressful condition. And then we have a bunch of images that are not so pleasant to look at, and these comprise our stressful condition. So when we repeatedly present these unpleasant images to people, we have this sustained exposure. It induces a mild stress response in participants. And there's lots of ways that we can check that we're doing this. One of these is by asking participants to rate, for example, on a scale between 1 and 9, how stressful and how unpleasant they find these images. And we do indeed find that they rate, that they feel more stressed following a series of these unpleasant images compared to the pleasant images. And likewise, they rate them as more unpleasant. So we know that this sustained exposure can increase subjective perceptions of stress. But we can also look at changes in physiology. So look at stress responses in the body. In my own work, as well as in other researchers, I'm not presenting this data today, but the sustained exposure increases neuroendocrine responses. So these are our stress hormones. It increases cortisol, noradrenaline, and adrenaline levels. It increases skin conductance. So if we were to record using an electrode from the palm of the hands or on the fingertips, we can measure the sweating response, and we see an increase in heart rate. So these bodily or physiological responses are not too different to what you would expect, getting sweaty palms and a racing heartbeat watching a scary film, for example. So these measures here are telling us something about sympathetic nervous system activity. And put very plainly, the sympathetic activity or the sympathetic system is the one that drives our fight or flight response. So we have these changes to try and prepare and mobilize our body, including our muscles, for an impending action to make sure that it's appropriate to deal with that stress or that threat. So this could be to flee, it could be to stay and fight, or in some cases, it could be to freeze. So participants are performing this task, keeping as steady as possible while viewing these stressful and non-stressful images. And what I'm interested in knowing is, well, how does your ability to control force on this level, on this target level, how is it impacted by what you're seeing, by the emotions that you're processing and by the continuous presentation of these stressful images? And then what brain regions might be involved in this simultaneous emotion processing and generation of a motor response, the grip force. So one of the measures we can look at is simply taking an average of force, how hard people are squeezing. We take an average over many trials, we combine the data from all our participants together, and we can plot the average force between the stressful and the non-stressful condition. So what we find is when people are viewing a series of these unpleasant images, that they produce more force. They're actually pressing harder on the force device during this acute stress exposure. When we ask participants whether they're aware that they're doing this, the answer is always no, but this is what we find. So this is quite interesting. We have this unique effect of acute stress on our motor system, on our movement. So then the question becomes, well, what's happening in the brain during this task? Are there specific regions in the brain that might be involved and that might explain this effect, the fact that they're pressing harder? And we do find a number of key brain regions that are involved. So on the next few slides, I'm going to show you the data from the MRI, from the neuroimaging, the scanning. when we have our neuroimaging results, we take brain slices and we can look at the brain from various angles. So front on, from the side, and from top down. So that's what you're seeing here. The same area of the brain from 3 different orientations. These yellow blobs here tell us which areas of the brain are more active during the stressful condition versus the non-stressful condition. So of course, when we perform these tasks, there's a whole host of brain areas that are active, that are communicating with each other. What we're focusing in on here is those regions showing more activity during stress. So it's a direct comparison. So the first region that we find is the right inferior frontal gyrus, shown in the darker red here. So this is at the very front of the brain. It's on the right-hand side. Of course, we've got the similar area on the left-hand side, but we found a specific increase in activity on this right-hand side. This is in our frontal cortex, which is where we have all our executive functions. So this particular right inferior frontal gyrus that you can see in these blobs here plays a really key role in attention, which is one of these higher order cognitive functions. And of course, in this task, participants are attending to the pictures, they're attending to what they're doing with their grip force. But not only that, it's taking in all of that information and figuring out, well, what's relevant, what's salient, what do I need to know in order to respond accordingly? So it filters all of that information and then signals to the motor cortex, which sits just behind it in here, what type of motor response would be appropriate in that situation. So again, this is coming back to, should I be fighting? Should I be fleeing? Should I stay on the spot and freeze and hopefully no one will see me? So it's a way of dealing with that impending threat, just like the animal kingdom does. The other key region that we found associated with the stressful condition, so higher activity, is the amygdala. So the amygdala is this little almond-shaped structure that sits in the middle of our brain, and you can see it on my results here. And this is part of our limbic or emotion processing system. So there's lots of research now showing that the amygdala processes both negative and positive emotional information, but that it responds more, it's more active for negative emotions, especially threat-related signals. So this very much ties in with our study that, with the finding that the amygdala was more active when people were squeezing harder when they're viewing these stressful images. And finally, we found significant activity in the cerebellum. So this is the cauliflower-shaped structure that sits at the back of the brain. It's really critical for our motor control, plays a key role in coordination and timing of muscle activity, and really important for precision grip force. And the particular part of the cerebellum that we found to be more active was towards the midline. And as the animation comes round, it's this middle part of the cerebellum. And this particular part of the cerebellum is also referred to as the emotional cerebellum. So this is also really fitting that we found more activity in this particular region for our stress condition. So it's always really great when you can link biology with behavior. That's always the ultimate goal of having neuroimaging, for example, with changes in function in behavior. So here we're linking changes in brain function with this, the fact that people are pressing harder on the force device. But what's really nice about this type of research and these types of questions that I'm asking about interactions between limbic or emotion systems and our motor control system is that it lends itself really well to investigate in clinical populations like Parkinson's disease. So before I talk about my next study in Parkinson's, I'll just provide a little bit of background and context about stress and what we know in Parkinson's disease. So there's a growing body of research testing this long-standing notion that stress plays a key role in Parkinson's. And it's really interesting that stress has been one of the earliest proposed causes of Parkinson's. So going back 130 years or so in the late 1800s, at the time of Charcot and William Gowers. Already back then, and this is one of the original drawings from Gowers, he wrote that prolonged anxiety and emotional shock are the most common antecedents of Parkinson's disease. But it's only been now really in the last 2 decades, since the early 2000s, where research in animals has shown that acute and chronic stress can profoundly influence the functioning of our movement system, especially for skilled motor behaviors like reaching, grasping, so our grip force. In terms of what might be underlying these effects, one possibility is that stress alters movement due to changes in dopamine, or changes in the dopamine, or changes in the functioning of the dopamine system. So dopamine is a neurotransmitter, it's a chemical messenger that provides signaling between our brain cells, our neurons, and it's really crucial for normal movement. It also plays a role in our fight or flight response. So this work that's been conducted in animals has led to this idea that an acute stress exposure leads to a temporary reduction in dopamine. So when we have this dopamine deficiency, which we know is important for normal motor control, when we're lacking in it, then we have this disruptions to our motor control. And dopamine, of course, is already compromised in Parkinson's disease. So in Parkinson's, we have cell loss or neurodegeneration of neurons that produce dopamine. And this lack of dopamine gives rise to all of the motor impairments that we see in Parkinson's. So on top of that, if we have individuals with Parkinson's who are already deficient, and then they're exposed to these stresses, which may further exacerbate dopamine deficiency, then we can see how stress may be a key factor in influencing Parkinson's symptoms. So overall, these studies have very much expanded the classical view of stress, that it mainly affects cognitive and emotion functions, to this idea that it's much broader than that and that it significantly impacts our movements. That acute stress may have the potential to accelerate cell loss in the brain and therefore accelerate neurodegenerative processes. That it has the potential to unmask motor symptoms at an earlier phase in the disease, in the trajectory of the disease. So we may have an earlier onset. And then it may lead to a worsening of the motor symptoms once the disease has been diagnosed. So taking all of this together and using what we know from the animal research, we wanted to examine in human Parkinson's disease whether acute emotional stress has the potential to exaggerate these impairments that we're already seeing in grip force in Parkinson's. So this study essentially replicated my first one in the university population, the younger aged adults. It wasn't in the MRI environment, it was just in the regular lab, but we included measures of muscle activity. So in this study, we tested a group of individuals with Parkinson's, and they were tested off their medication. So what this means is we asked them to forego their morning dose of anti-Parkinsonian medication, their levodopa, which normally acts to restore the levels of dopamine. And so here they came into the lab already in a dopamine depleted state. And we do this to try and tease out some of those motor impairments. And their data were compared to a group of healthy controls. And what we mean by this is a group of individuals without neurological disease, and they're matched in terms of age and gender. So just like before, they're asked to squeeze on this force device, try and keep it as steady as they possibly can on that target level while viewing these pleasant and unpleasant images. And the question remains the same: how is their ability to control force impacted by acute stress, and is it different in our patient population and in people without Parkinson's? So the results from this study, I'm not showing the bar graph anymore, I'm just showing the actual traces from the force device. And this is the data from our healthy control groups of people without neurological disease. So here we've got our trial of 12 seconds long, and they're increasing their force, they're squeezing on the force device and trying to hold it steady. In the blue trace here, we see the force from our pleasant or non-stress condition, and in the red, we see our stress condition. And you'll notice here this difference in force towards the end of the trial. So just like in our first study, in the younger age adults, participants produce more force, so they press harder while viewing these unpleasant images. So acute stress influences the amount of force that people produce. Data for the Parkinson's group told quite a different story. So here we didn't find an effect of stress on force. So there was no difference in how hard they pressed between these two conditions. But what you might notice here is that these lines are much wigglier for people in our Parkinson's group. So they find it much harder to keep force steady on that target level. They're much more variable, or you can think about it in terms of a decrease in steadiness. But what's important to point out here is that we only see this for the unpleasant condition. Down the bottom here are the results for the muscle activity, where the higher the spike, the more active the muscle, so the stronger the contraction. So the first thing you might notice is that people with Parkinson's disease have more muscle activity overall than people without. And this is due in part to rigidity. So this is one of the key features of Parkinson's, where they have stiffness in their muscle. It's in a state of higher contraction, if you like. But again, what's the really interesting part is that muscle activity is higher still for the stressful condition. So what we can conclude from this study is that acute stress affects grip force differently in people with this movement disorder. It doesn't affect how hard they squeeze, but it affects how variable they are at holding that squeeze, and it affects their muscle activity. So overall, this supports this idea that stress profoundly influences skilled movement like grip force. And remember, we need dopamine for normal motor control. These individuals came into the lab off their medication, so in a dopamine deficient state. So we expected some degree of impairment in their grip force control. But on top of that, we see this additional exaggerated impairment in the stressful condition. So it's possible here that this acute stress did indeed, like we saw in the animal studies, lead to further dopamine reduction, and this might explain those exaggerated movement impairments. So in the next study that I'll talk about, I moved away from looking at skilled movement, so not looking at grip force any longer, to specifically looking at one of the cardinal features of Parkinson's disease, and that's the Parkinsonian resting tremor. So these are the involuntary movements that we see in the upper limb or in the lower limb, the legs. We can also have resting tremor in the jaw. So these small oscillations when someone's at rest. So we wanted to know, similar vein, how might emotional stress be influencing or exacerbating that tremor? Because we've had a lot of anecdotal evidence in the lab, in the clinic, that indeed it does, that when people are really stressed that week, they report their tremor is worse. If they've experienced a negative life event, their tremor is worse. But surprisingly, there hadn't actually been any studies specifically looking at this effect, examining whether emotional stress does indeed exacerbate tremor. So in this study, we tested 34 participants, and all of these individuals with Parkinson's disease had rest tremor in their upper limb. And this time they were tested on medication. So they came into the lab following their usual medication regime. There was a range of symptom presentation. So some people had a mild tremor, others had quite a severe tremor. And we use clinical rating scales to assess their tremor at the time of the testing in the lab. As indicated in the photo here, and as I alluded to earlier, we have an accelerometer taped over the index finger to measure those movements in the finger associated with those tremor oscillations. And we record the muscle activity from the forearm flexor muscle. So this is the muscle in the forearm that flexes those fingers. So we expect to see it active or more activity when the fingers flex, and quiet or at rest when the fingers relax again. And for all of our participants, we measured tremor on the left or right hand, whatever one was most affected. So when we're looking at resting tremor, it's really important that the forearm is supported by a table and the hand is dangling freely, because we know that any activation of those muscles or any movement that the participant produces can diminish that tremor. So they're sitting there, we ask them to let their tremor occur as naturally as they can. Of course, it's in a research environment, so it's a little bit odd. But they're doing this while they're watching the screen. And again, we're presenting a series of pleasant or unpleasant images. And their only task is to simply watch these photos. So on the next slide, I'll present data from the accelerometer and data from the muscle activity. So this is just some example data from one participant. So this gentleman had tremor in his right hand. It was considered to be of moderate severity, and he'd had tremor for the last 15 years. So this is a snippet of data, just 30 seconds of recording from the accelerometer in the top trace and from the muscle activity in the bottom trace. And it's a little bit hard to see here, but if we zoom in just on a 3-second period, the individual tremor movements become a little bit more obvious. So we can see these up and down movements of the finger, or the accelerations and decelerations that are being recorded or detected here. Every time that index finger is moving up and down, or flexing and extending. And accompanying that, we can see this bursting like activity in the muscle. So every time it flexes, we see an increase in muscle activity, then it relaxes, it bursts again and relaxes. So this data here is from our non-stress condition, when they're watching a series of those pleasant images. When we look at data for the stressful condition, I think it's pretty clear here what we find. So again, it's obvious even in the 30-second period of recording, as well as in the 3-second period of recording, where we see this marked increase in tremor. So tremor amplitude, or the amount of movement in the finger, and in the amount of movement, amount of activity in the muscle. So I mentioned this was data from one participant, but this very much reflected the results when we combined all the data from all of our participants together. So from this, we conclude that tremor was exacerbated by acute stress. And indeed, this is just through watching these emotional images. And this is very much in line with what patients are telling us in their everyday lives. And again, highlighting this close relationship between our emotion and motor systems. In the last study that I'll talk about today, it's a little bit different to the rest. It's not a lab-based study, but we conducted a survey study during the first major nationwide lockdown due to COVID-19. So this is 2 years ago now, March and April of 2020. We were all at home. My research in the scanner and the MRI machine was taking a break because we couldn't see anybody. So we conducted this questionnaire study where we administered online a group of questionnaires to almost 200 of our participants that are part of our longitudinal study in Parkinson's disease in Christchurch at the New Zealand Brain Research Institute. So the questionnaires went out to people with Parkinson's as well as people without. So at this time, March 2020, there had already been a wealth of studies looking at the impact of the nationwide lockdowns. Of course, New Zealand wasn't the first to be going through this. So these studies looked at mental health and well-being, among other concerns. And also at that time, there'd been a number of studies, very small case studies from overseas, looking at people with Parkinson's disease who had been infected with COVID-19. And these showed that the infection did worsen their motor symptoms. But given our incredibly low infection rates at that time, we thought it would be interesting to examine the impacts of lockdown without the presence of infection. And here we were interested in people's perceived stress levels, and for the people with Parkinson's, whether they had any self-reported changes in their Parkinson's symptoms. So I mentioned we sent out a whole host of questionnaires. I was primarily interested in 3 of these. So the first was the perceived stress scale. This scale has 10 or so questions asking about people's perceived stress levels. You score the questions, you tally the score up, and it can provide an indication of low, moderate, or high levels of perceived stress. We asked people about their physical activity levels during lockdown. Of course, we could still go out for walks and so forth, but I was asking about the intensity of that physical activity and the duration. And then for people with Parkinson's, they filled out an additional questionnaire where we assessed how they felt their symptoms might be changing, whether they noticed any changes, was it better or worse during lockdown, and what was the magnitude of that change. So first, looking at the perceived stress scale. So here the scale goes from 0 to 40, where the higher the number, the higher the amount of stress or perceived stress. And we did find that our group with Parkinson's disease, on average, felt that their stress levels were higher than our people without neurological disorder. But what was quite interesting here is that these levels were considered relatively low, so an average of 13. And this was in stark contrast to other research that was being conducted around the world using this exact same scale, particularly in Spain, Italy, Mexico, and India, were some of the big countries already reporting on their lockdowns, where they were looking in their general population, so not necessarily with movement disorders or neurological disease, they were reporting moderate to high levels of stress. So we thought this reflected our relative success of our lockdown back then at containing the virus. So although stress was higher in our patient group, when we combined everybody's data together, 70% actually felt that their stress levels were the same during lockdown compared to prior to lockdown, and 21% felt that their stress levels were higher. Regarding the Parkinson's symptoms, we were interested in a number of symptoms: tremor, slowness of movement, any difficulties with balance and walking. And I'm just reporting the results for tremor here because they were very similar findings. So from our 134 patients with Parkinson's, they self-reported, 90% self-reported they had tremor. And 30% of those individuals thought that their tremor was worse during lockdown compared to prior to lockdown. And this ranged from a slight worsening through to a severe worsening of tremor. So the next question we wanted to ask was, well, of those people that felt their tremor or their motor symptoms were worse, was there an association with their perceived stress levels? And we found that there was. So those people reporting that their tremor was worse, in the light blue here, their perceived stress levels were higher than people that felt that their symptoms didn't change. So not only was stress higher in patients compared to controls, it was higher in people that felt that their tremor or their motor symptoms was worse. Again, providing supplementary support for my lab-based studies, but for this close relationship between stress and motor function. So just to finish off, movement disorders like Parkinson's are multifactorial, but so is stress. It can exert its effects on a variety of systems. And my work has shown that it not only affects emotional and cognitive processing, but that it profoundly affects our motor system and our movements. Acute stress can exacerbate motor symptoms of neurological diseases, and I'm focused on Parkinson's disease here. But in talking with other people, we know that it's also affecting a variety of neurological disorders. Of course, we can't avoid stress, nor can we prevent it from influencing the course of neurological disease, but I really think we need to be mindful of its effects and the potential for acute stresses to compound on top of each other and become chronic. And we also know that dopamine seems to be playing a role here, and this is something that my current research is looking at. I'm still working through the results of that study. So I think when we're developing new, or importantly, individualized therapeutic strategies, that we really need to be considering people's stress levels. We need to think about their current emotional state or their current mental health. Because stress and emotion, I think, certainly have the potential to determine the success or how we might respond to treatments, common therapeutic avenues to manage movement disorders. And perhaps complementary interventions could be focusing on reducing stress. We could think about stress resilience, or providing a way to cope with these stresses better. Improving mental well-being, whether that's through yoga or meditation, for example, whatever it may be for you, getting out and going for a walk along the beach. We know that if we reduce stress, then this can assist in enhancing our motor function as a really viable, non-invasive strategy for movement disorder populations. And this is something that I'm hoping to investigate in the future. So with that, I'd like to say thanks for listening, and particularly acknowledge my colleagues in Geneva, in Christchurch, and Maddie, my research fellow in Dunedin here. So